Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
It's my pleasure to introduce Dimitri Samaras. He's now associate professor at Stony Brook, and he's been there for about 7 years, right? He did his PhD at University of Pennsylvania with Dimitri Metaxas, and back then his interest was in modeling faces, like computer graphics and so on, which stayed his interest for a while at Stony Brook as well. But now he started doing something much more interesting, which is what he's going to talk about today. I'll just let him continue.
Okay, well, thanks, Nebojsa. Thanks for having me here. It's a great pleasure to present work that we've been doing the last 2 or 3 years, mostly a collaboration between me, my graduate student Lei Zhang, whose thesis is the bulk of this work, and Rita Goldstein in the medical department of Brookhaven National Labs. There's a number of students that are starting to join this project, and also other neuroscientists that are helping us. So the idea is that we want to discover patterns in space and in time in fMRI data. And I'll just give a brief description. This is not particularly scientific, but hopefully it'll give an idea. So what happens is that when blood goes through a certain region of the brain, that means oxygen goes through there as well. And the magnet can actually image that oxygen. So blood oxygenation levels, this is what fMRI is measuring, and you end up with these images here, with these hotspots, let's say, which are show brain activations. There is no clear understanding yet of exactly how this relates to neuronal activity. So people don't know exactly what they're imaging, but all they know is that these regions of the brain seem to have increased blood flow. So now,
we also know that in general, when you look at something, some visual areas.
Yes, but there is no physical model that will say this is the physics of the neurons, sure, chemistry, and this is how this will translate to blood flow. So exactly what it is that you're measuring, it's not exactly.
Yeah, but it's rough.
It's rough. It's rough, and it's been used a lot. There have been a lot of studies until last time I looked at this statistic. My collaborators were telling me that in the first 10 years of fMRI, there have been at least 17,000 fMRI studies that were done. So usually the neuroscientists that do this kind of studies, they start with a hypothesis. Okay. They want to see which region of the brain corresponds to a certain activity. They think that particular region of the brain is involved in a certain activity and so on. What they do, they collect the data, they run a t-test, basically. Confirms the hypothesis, doesn't confirm the hypothesis, that's it, they go publish it. But what's a little bit more interesting in terms of, I guess, the group of people that are in this room, is what is it that you can actually see in this data? What is it that you can actually find? And one can define a number of pattern recognition problems. One of them would be, for example, to be able to figure out what the brain state of an individual is. Was that person reading a book? Was that person listening to music? Was that person chewing gum? We don't. That's something that you might want to find. Usually that's relatively straightforward in the sense that it's different areas that activate. A slightly more complex problem, and this is the one I'm going to be talking about mostly, is how do we actually separate different groups of human subjects based on fMRI data. In this case, the activations are going to be in the same regions, more or less, but they are going to be of different levels. Why? Because function is somehow changed. The data set that we're looking at and the groups that we're looking at are cocaine addicts versus control subjects. Similar studies are being done for people with Alzheimer's. Similar things are being done with people for people with schizophrenia. So this.
So again, what do they traditionally do with p-values? With tests? You said traditionally they would compute.
Just a t-test, yeah.
On what?
On whether they started with a hypothesis. This brain region gets activated when you do this.
Region.
Yeah.
And then what do they do with multiple hypotheses?
I don't. They will run different tests for each one and see where there is significant statistics. Basically, that's about it.
And they have to correct for all the hypotheses they made.
I guess they'll do. They'll get. These are psychologists. They know ANOVA. Okay, that's where the story stops with them. So we actually used some of the stuff that they found this way, as I will mention later. So this is the problem.
You're saying that you want to split them, but what are they going to do? What's the measurement going to be?
I'm going to come to this. So now let me just try and convince you that you need to sit here for an hour and listen to this. Well, it has to be a hard problem, right? So why is this hard? First of all, this data is really hard to collect. It costs a lot, both in terms of the manpower that you need. It costs a lot because you need the specialized scanners. It's human subjects research, basically.
They survive after magic, right?
They do, yeah. The one thing that I think my collaborators are not allowed to do is to test models of what happens to the brain when someone's becoming cocaine addicted. They're not allowed to give people cocaine. Okay, but they do have to go and find cocaine addicts. They have to work with treatment groups and recruit and so on. And usually to run a subject, it takes about 2 hours if everything goes well, because they need to take all sorts of other interviews and figure out, what kind of state they are, when's the last time they used, and so on. So this is, in general fMRI studies, there are very few studies where you will find, for example, 200 subjects. Okay, most of them, especially the ones that are done in university settings, are in the order of a dozen subjects. Okay, the studies I'll be talking about are we had up to 40 or 50 people. I'm going to discuss 2 different studies. So, but then you have not that many subjects, but the data itself is fairly high dimensional. Just and someone can do the math here. It's about in the order of 250,000 voxels at 87, and for the particular task that I'll talk about, it's 87 scans. Let me just say, for those who don't know this, that we're getting 1 scan every 3 seconds. And now you have to deal with increased variability. I will talk about inter-subject and intra-subject variability, meaning that if you get the same subject to perform the same task, it's not going to be exactly the same. Either because half an hour has passed and they're more tired or more awake, but also there is task habituation effects. So they repeat the task, and as they repeat it, they become more used to it. Brain function changes, and then there is the variability between humans. And and then the last challenge is that these changes are relatively subtle. Okay, you can't really see these things that easily, but just by getting a brain scan. Things like Alzheimer's and so on. Yeah, of course, the brain shrinks. Okay, the brain dies. You can really see these things. Here, it's much more subtle. The data in general is noisy. So in your typical fMRI study, you're talking about a signal-to-noise ratio of 5%. And for that reason, the standard processing, they use to toolbox called SPM. It runs over MATLAB. This is the standard in that community. We use the same thing so that we can actually be able to compare with other people and make sense to them, and kind of have the same biases. So they regularize and normalize the data to death in order to be able to get something useful. One common thing is that they create what's called contrast maps. And the first things I will talk about are experiments on contrast maps. Contrast maps do what? They average, basically, all the scans. So they lose all the temporal information. But at the same time, activations, if there was a region that was consistently activated over a number of scans, these things are going to come through.
Interpatient or across all the patients?
No, it's per person.
Per person.
Per person. And you can actually choose which contrast maps you will create. And I will be discussing these different contrast maps. It could be per person, and it could be per condition, per experimental condition, or you can break up the scans in any way you want that is meaningful. And then get all the scans that refer to the same condition and average them. That's these are contrast maps. This is post-processing.
So you measure, and then you can say, I want this, I want that.
Yeah. And I will discuss actually how different contrast maps lead to different results, which is not a bad thing, as we recently found out. So, just since I come from a background that uses images, I have to show something. Okay, so can you tell what is the difference? How can you say which one is the cocaine subject brain versus the control subject? You can't really see very obvious patterns in general here. I don't even remember, since I've made that slide, which one is the control and which is the addict. Anyway, so we've done work in 3 ways. First of all, the first thing that we did was the so-called spatial analysis, contrast maps, which are basically static images that you get. But then we really didn't like the fact that we're throwing, together with the baby, we're throwing away the bathwater. Or together with bathwater, we're throwing away the baby. So, so we want to keep the bathwater, okay? So we're losing all the temporal information. By doing this averaging, we can't really see what's happening with, for example, people being faster or slower or having different temporal patterns. And so we wanted to do this next. And then once you start looking at the temporal domain, then you realize that these areas of the brain that seem to activate, they are not independent. Okay, these are not independent time series. There is dependencies. I will call them functional connectivity. There is a little bit of a nomenclature fuzziness here in the literature, but I'll call them functional connectivity. This means that we're just looking for statistical measures of connectivity. There is no physical evidence that there are neuronal pathways from one region to the other that are actually active. Okay, just so that you guys are clear on this. So, spatial analysis, it has been done before. I think the first one was for Alzheimer's. Then they started looking at classifiers to detect cognitive states, meaning memory versus looking at something versus reading and so on. In our case, as I've said, the cognitive effects are fairly subtle, and we're talking about the same general brain regions for both groups.
So for the state, that's an easy problem? Like
It's
cognitive state?
It's an easier problem. It's an easier problem in the sense, okay, I'm going to skip the temporal analysis. Functional analysis, The important one to remember is the dynamic causal models. This is something that has a lot of traction in the neuroscience community. But I'll say why that was an easy task. So we started looking at this as a 3D video understanding problem, which was, I guess, an okay initial approach. It will turn out that it is similar to 3D video in the sense that you have 3D data that changes over time, but it is not similar to 3D video. It's the wrong analogy in the sense that you do not have spatiotemporal gradients. Okay? You don't have things moving from one voxel to the next. You have just a constellation of light bulbs going on and off, if you want. So we tried to do this as general as possible, and at the same time, we hope that at some point this will lead to clinical applications. So now, let's talk about the experimental data. In the experiments that we did, I'm going to talk about two experiments. One is the one where we run everything. The other one is under collection now, and we're just finishing with processing. So for the second experiment, the data is still kind of preliminary. The subjects perform a very simple go, no-go task. That means they have to press a button. They're shown two different images. If they see image one, which is some kind of a red image, they press the button. If they see the green image, they don't press the button. Okay? It's an easy task. But before they do this task, what the neuroscientists care here is not whether they can press the button or not. Okay? What they care about is the sensitivity to reward. So before they're shown the image, they're shown a number. And that number is how much money you're going to make if you do it right. Okay? And this is real money in the sense that you do it multiple times, and if you do everything right, by the end of the experiment, you go away with $55. Okay? For spending an hour in the scanner.
Yeah, that's the question.
Exactly. Apparently, that was enough to get. It was easier for them to recruit cocaine addicts than control subjects. And I have to say that chances are that I haven't asked them. I haven't figured out how many of the control subjects were graduate students, which might invalidate the experiment. Okay.
Or heroin.
Graduate students. So there is three conditions, three monetary conditions. So when I'm talking about conditions now, I will mean the monetary condition. 0 cents, 1 cent, and 45 cents. Okay? And the expectation is, so what we're looking at is actually the areas in the brain which are associated with reward processing. Now, reward processing for neuroscience is the major system, because that's what makes you get out of bed. That's what makes you come here and listen to this talk and so on. And for cocaine or for drug addiction, that's what makes someone decide whether they're going to go into therapy or they're going to go look for their next hit. Okay? Which is, again, important in terms of clinical intervention as well.
Very quick question, Sir Devil's Advocate. You're trying to separate cocaine addicts from the controls, and you're basically doing this on base effectively of the blood flow in the brain.
Yes.
And if I asked you whether you could actually, if you trained your model and everything based on the blood flow in the muscle, would you be able to separate these guys as well? Maybe you're making a problem separate. There might be more difficult tasks, but for this particular task, is it clear that the cocaine is not impacting their blood flow in general, rather than the operation?
They have, they do. From what I understand from the people I work with, they have actually control for these things. They've checked that the physical condition is pretty much the same.
Okay, so the brain is basically
and the people that do this are not under the influence. They might have used the day before or a few hours before, but when they go into the scanner, they are not under the influence.
And also, it's clear that you're not separating rich people from poor people, not opposed to cocaine addicts to non-cocaine addicts?
They are, they are taking some care to, so when they recruit subjects, they do get a full background information and so on in order to recruit someone. So they're taking some care to have groups that are of similar socioeconomic status. But yeah, that's a good point.
So then you can actually see reaction to these things in oxygen level in blood.
Yeah. So basically,
it's that fast. The change in oxygen level is that fast.
well, yeah. I mean, if you these tests happen they keep repeating within a few seconds, right? Within a second or half a second, and so on. So it's enough time that the whole thing, see the slide, see the image, press the button, is within the 3-second cycle.
And that is not too long. I mean, you don't make a decision much faster than that when you see this. I mean, I assume that it's all gone by the time. You don't even require new oxygen coming in to make a decision.
No, it actually, yeah, there is, blood goes to your brain.
You actually see that it's that slow?
The way they designed the experiment, so they have this particular experimental design which is called block design, which means that things go on the clock. And I have a slide somewhere, I've taken this out, where I have the exact timings. So it is half a second for this, half a second for that stage of the experiment, and so on, and everything is absolutely timed. So they can see these things. Yeah, and this is one thing that I'm going to come back, keep the block design thing in your minds, because a more interesting thing would be, what do you do when you just show people movies, okay? But then you lose that exact timing and gating type of thing. So anyway, so what we're testing here is sensitivity to reward. I don't know. People are doing, psychologists are doing fMRI studies with movies just to figure out what's happening when you see a scary movie versus a happy movie. And
I'm just wondering how repeatable that is. I mean, do you see any kind of correlation?
People report results. Now, people report results. I can't really tell you. And there is a community that accepts them.
No, actually, neuroscience fMRI is very good right now.
Yeah, it's
like Nature Neuroscience is like every third cover is about an fMRI study.
Sure, but what does it measure? Then we're trying to get a better.
There's a big debate on what it measures.
yeah. So anyway, so yeah.
The resolution is improving, right? Like
Resolution
is improving.
Well, up to a point, not really. I mean, they need to come up with stronger scanners. FDA is actually, I think, in the process of approving stronger scanners for humans. Okay, the resolution is really improving for mice because they put them in 21 Tesla scanners. People for clinical, they go to 1.5 Tesla. For experimental, they will go up to 3, 4.5. That's where. But I think they're in the process of going up to 7 or 9 Tesla, FDA approved for humans. Anyway, so yeah. So now what I'm going to talk about is 6 contrast maps for each run. We are checking each experimental condition, its monetary condition versus baseline. Baseline means you do nothing. Okay, this is just the noise in your brain. And then we are comparing the conditions with each other. Okay, so 45 versus 1, 45 versus 0, and 1 versus 0. Just to see the differences in activation. And before I go to this, what has been observed already is that it seems that when it is 0 cents, the reward system is not really going to activate. When it's 45 cents, the reward system is going to activate, both for controls and for addicts. Okay.
So they know ahead of time that there's going to be 0.
They're shown the slide. They're shown it before each task. They're shown a slide.
But that's not kept constant all the way through.
No. You alternate and you're seeing, and they have designed it so that for a specific amount of time you're shown 45, then you're shown.
So you can have some spillover from the previous.
They gate it so that that's the whole block design, so that and when they publish these things in the methods, they will actually spend 2 pages describing exactly how they've done it so that it's a valid measurement. So what they found was that the 1 cent condition, that's where there is a big difference. Okay? For the non-addicts, activations corresponding to 1 cent are pretty close to, are very similar to the ones for 0. For the addicts, they light up. Not as much as 45 cents, but they definitely light up. We don't know. It's not clear if that means because that for them 1 cent is still money. It gets them closer to the next fix, or or the reward system is actually broken.
So with cocaine, but you become better with money when you start using cocaine.
No, you become worse with money, because you cannot really tell the relative differences in reward. These are people that will happily give you their car for their next fix, right? They can't tell that if they go sell the car to a dealer, they will be able to get much more money, many more fixes. They can't deal with this. So, and it's unclear if the reward system was already problematic before, and that's why they became addicts, or
Is anybody planning to do a study like that?
you can't give them cocaine. That's the problem.
You can track
Hmm?
them. You can track them.
So now they're doing
That's why they're doing HIV studies.
now they're doing longitudinal studies where they've started getting the same people coming back after 6 months in treatment or after a year with no relapse, just to see whether.
For people that are at risk of becoming cocaine addicts before they become cocaine addicts.
I'm not sure that it's easy to define such a group. Hmm?
That's what they do for HIV.
Yeah, but the HIV studies, it's specific populations. I don't know if they have that for cocaine.
Hollywood stars.
Yeah, exactly. So, so this is the let's say the physiological knowledge that we started with. So now let's see whether we can actually use this. And this is, as I've said, we're trying to do this as general as possible. So we started with PCA. Okay, you have to start with something. And people who were doing mental states were actually able to get good results out of, say, SVMs or GNBs and so on. They were getting decent results and saying whether you're reading a book or you're looking at a picture or you're thinking of a memory. In our case, these methods, as I'll show, didn't really work. Okay? This worked. So boosting worked quite well, and most of the results I will show are on boosting. We actually, given the fact that this was an experiment where they actually did different repetitions, and they were broken into 3 different blocks. Blocks in this case means 3 different sequences of showing these 4501 slides, and just to make sure that it is random. Okay, and then there is a repetition. So for each subject, you can say that you have 6 data sets total, which means that we can exploit this for our classification. So just to show some numbers, things are starting to get. So if you just compare to baseline, things are pretty bad. Once you start looking at different,
What do these things mean? T1 plus 1R?
these are the different blocks, T1, T2, and T3, and 1R, 2R, and 3R are the repetitions of those blocks, which again was the design of the experiment. Hmm? So percentage of classification when you're comparing to baseline is pretty low. It's in the 50s. When you
What was the baseline?
baseline is when you're not doing anything. You just sit on the scanner. Okay, this is your standard, let's say, brain activity at rest.
So you're trying to classify whether it's a 1 or a baseline or 0 or a baseline or 45 or a baseline?
No, we're trying to classify, to say when I'm looking at the difference of activation between 45 and baseline, can I can I classify cocaine addicts versus control subjects? Okay.
So those are contrast maps, effectively.
So these are all different contrast maps. Okay. And as I've said, you can define your own contrast maps. And so these are the more interesting ones.
Same number of cocaine and non-cocaine?
Approximately, I think there was in this study they had about for the numbers that I have here, which are approximately 2 years old, they had about 15 cocaine addicts versus 13 non-addicts. And
52 is roughly chance.
yes, and the way this is the same for all the classification results I will show. It's leave one out cross-validation. Okay. So.
So why do you say this didn't work? You're getting a significant result?
So we're start for specific blocks, but once you start averaging everything, things start falling.
But it's still better than chance.
Yeah. This is the condition that we're talking about. The activations when you start looking at the one condition.
45 versus 0.
Again, this is you're starting to get some good things at this point. 5 nearest neighbors are starting to do okay.
Out means that you're leaving the subject out?
So we're training because we don't have enough subjects to get a good split between training and testing.
So you're leaving one subject out?
For each subject, we train on all the rest and try to recognize that subject. And we do so. We had a total of 27 subjects. That means we did 27 classification experiments. Okay.
So how would you graphically say 5 nearest neighbor? Does it mean 5 different areas of the center points for 5 different areas on the brain? The centers of the 5 nearest neighbors.
Yeah.
There are 5 different areas of the brain.
Mm-hmm. Which are the most the 5 nearest neighbors in terms of discrimination.
Wait, 5 nearest neighbors, full boxes of your voxels?
Hmm?
Your mean, your center is a full box of full set of voxels.
Yes.
You have 5 of those.
You get 5. The 5 of in the data space
Right.
that we are the 5 that
Right.
5 nearest neighbors for the 5 voxels that are closer for classification to whatever the.
Right, so full set of
We actually-
vector voxels.
Yes.
And 5 of those. So the
Yeah.
full brain.
We tried a number.
Wait, isn't it 5 nearest patients?
Hmm?
Isn't it 5 nearest patients? No.
It's looking at.
By PCA.
No, it's
No, no.
each image, each contrast map is labeled. This is from an addict. This is not from an.
Sure. But
Yeah.
when you say 5 nearest neighbors, it's not 5 nearest neighbors in the map. It's 5 nearest maps.
Yes, 5
Yeah.
nearest maps. So
5
that's
nearest
not
Yeah.
maps by
necessary.
PCA projection.
Yeah.
Yeah, it can be from the same subject.
So 5 nearest subjects.
Yeah.
Right. Do you have
So-
one or more? That's a good question. Do you have one or more scans per subject?
What do you mean one or more? We
So
have.
5 nearest neighbors could be all from one subject, or you had one scan.
Well, you get one contrast map.
one per subject.
Yeah.
So it's 5 nearest subjects, and then
Yeah.
you look at take the majority vote.
Yeah, which we'll come back to this majority vote. So we tried a number of things, whether we take the full vector of the voxels where we find the most active regions of interest. Usually you don't look at a voxel only, you look at the neighborhood of 8 voxels just because there might be slight registration effects.
And that goes to correspond to the regions of interest, meaning the reward stuff, or is it? I mean, does it have
This-
biological interpretation? These 40 best ones.
These ones we don't. Actually, we're still looking at this. No, this is just the most active.
Right. Okay.
Okay, and then for Adaboost, we're actually starting to do a little bit better. We're reaching 90 for specific for individual blocks.
So can you comment on why it's falling down on the averaging?
Because-
It's just what?
It's just showing some.
No, that means that if I test only on one specific block, which means for all the subjects, the sequence of the 45 zeros and ones is a specific one, then
Train on that and test on that.
then you get better. You expect to get better results. But once you start mixing things up, then I'll show you a little bit later. We've just done one.
So then you're saying that there is a
Spillover.
spillover from the previous
No. All I'm saying is that since things register in time much better, in the end, they're more directly comparable. The fact that there might be some crosstalk. This is noisy stuff, and you don't really know if you've been excited first and then by a 45 and then by a 0, whether that is different from getting a 0 first and a 45. So we're checking this, and this is kind of.
What are classifiers you using?
Just simple yes, no. I mean, can't remember which one it was. It was. Yeah, it's just basic decision stumps. One thing that we did, and I'll show the numbers a little later, we just tried this just to see whether it changes things. If you actually, instead of just mixing all of those things together, get the most discriminative, do the individual things, and get the most discriminative features of each one in the end. So if instead of having one set of contrasts, so here it's all of a contrast map that's created by all of those. So instead of having one contrast map, you actually do 3 contrast maps and get best features from all of them, then you don't have this drop in performance.
But you're modeling them independently.
So if you model them independently and you build a classifier that looks at all 3, you don't get this drop in performance.
Can you explain how you get the features for all of this? Are you averaging for the time block 1, 2, 3?
So here, all means that you have created a contrast map that has taken all the blocks together, averaged all of them. Whereas this one means that you've averaged only the T1s, only the T2s, only the T3s.
So block meaning block in time?
Block means a particular sequence of stimuli.
Of reverse,
Yeah.
So could you, they never tried just like taking 2 blocks that are identical and just summing specific time points within those blocks? It's too noisy. Like
That
a
is-
block has, I don't know, 100 trials inside, say. So you could take data point number 1 here and data point number 1 there, sum them up.
That we did a little bit on the temporal when we looked at the temporal information, but for the contrast map, no, we didn't do it.
And the features were still PCA?
Where?
You're still working on in the projection space? You still do PCA here? PCA feature?
No. This is the original.
Voxels,
Yeah.
Just the,
Yeah, but
this
then the weak
huge
learner
dimensional.
just chooses a feature or what?
Yeah, the weak learner.
It chooses one feature and thresholds on that feature.
Yeah.
So it's basically feature selection.
Yeah. So now, since one thing that we tried was how to, since we have these different classifiers now, you can build these different classifiers on the different blocks, on the different contrast maps. And there is this variability, okay? Some of those features that you will learn might actually end up for one block, they might tell you that someone's an addict, and for another tell you that they're not, okay? Why? Because Adaboost minimizes that way. So, but that's not something that you actually want to happen. So we just came up with a way to throw basically these features out, to inhibit those features. That's what we call boosting with side information. So the idea is that
Before you go, can you actually explain why you prefer intra-subject variability? You're increasing both variabilities.
you want intra-subject consistency.
Okay.
So you want the same guy to always be classified as an addict.
Okay, so it's a little like that.
That's so here is some voxels that are sometimes will tell you 1, sometimes they will tell you 0. You don't want these guys. Okay?
I'll tell you 1 in terms of classes. 1
Yeah.
would be cocaine or 0
Yeah.
would be control. Okay.
So in this particular case, you know that even if it's different trials, the label should be consistent. So that's just a way we came up with a fairly simple rule 1 and 1 line modification on Adaboost to just put that in. So and yeah, it did improve recognition rates or error rates in this case. And now let me talk a little bit about the second experiment.
Here's an experiment with crack. Get 50 cents.
So now, since the neuroscientists were pretty happy that yes, we can do classification, we can see these effects. Now, the next thing that they want to study.
So can I ask a question? So
Mhm.
when the medical people looked at the features chosen by your boosting, could they learn something about what voxels are important?
In this particular case, no, because there are too many. But I will talk about a different experiment where we gave them fewer voxels and they liked it, or they told us they did.
How many voxels did you end up choosing?
This one,
Like the first one.
the first,
Roughly speaking.
roughly, no, this is, I think it's we did 100 or 200 classifiers and slightly, a few hundred voxels. I was actually, I was preparing for this talk yesterday and I did call the student who's graduated and I said, give me the exact number. He said, I forgot. But we're repeating these experiments now and it sounds like it's, yeah, I've learned something from that experience. So we're repeating the experiments now and we're actually keeping this down.
Several hundreds.
Yeah, several hundred meaning 300-400. So, but it's not 10,000.
Okay.
Okay.
And still they think it's too much. Why do they think it's too much?
Because you don't have that many subjects. Oh, why the neuroscientists think? Because they can't really tell if you give them all these activations unless they really cluster. They can't really tell, these things in
They
various.
don't really cluster? I would expect that.
We're still working with them on this. I mean,
Of course, obviously, like the first few features should be very informative. Right?
yeah. So, and that's actually something we're preparing this stuff for a neuroscience submission, and this is exactly the stuff that we're preparing now for them. So, probably in a month I'll have much more information on this.
But you could probably test. I don't understand. You can't. Well, because there's many voxels, you can't basically say you can't test one against everybody and so on. It just basically does. But you could potentially impose the planes and basically come up to 4 or 5 planes that separate out 5 pieces of space,
Yeah.
where you can say, well, it this block and that's what data allows,
Yeah.
rather than actually going testing each voxel independently.
Yeah. So the second task now is more complex. Now they want to study both money and drug use. So a standard test is called the stroop test. It's a standard psychological test where you are basically shown something which is color coded, and you're supposed to focus on the color and not on the text. But it's kind of a confusing thing. They want to measure how things influence. So here, they are supposed to press the button when they see a yellow word. Okay, and they get words in yellow, blue, green, and red. So half of the words that they see are drug-related. Okay, high, crack, The other half are so-called neutral words.
Actually, it's quite nice that boosting works on this test.
So they want to, so basically they want to compare. So the drug cue is the really strong. They know that for the addicts, the drug cue is really strong. They want to compare that with a monetary reward. This data is still being collected, is still being analyzed. These results are very.
Compare with the monetary reward, compare with the other experiment, or in this experiment?
In this experiment.
They also have money reward.
There is also a money reward. Now they get 4 conditions: 0, 1, 25, and 50. And they are trying to see what's, and I can show some preliminary results where I can tell you this really. Probably if someone was to develop something for your employer to use before they hire you, they would go for the drug versus neutral. Okay.
But you could just do it without monetary reward. I mean, do you expect that addicts would make more mistakes?
Yeah, and it is true. They measure all this other data like speed of response, number of mistakes they make, and they get. There is definitely changes in behavior.
And they get a word if they guess the color, right?
If they press the button when they're expected to press the button.
If the word is yellow.
And they're expected when it's yellow.
Yes.
They get the yellow. If
Yes.
they press on yellow, they get the.
And if they
Yeah.
don't press, they get as well.
And but they press. What happens? And when they see a drug-related word, they press anyways, even if it's not yellow.
They might make more mistakes. Yeah. They might press anyway, or they might or they will press faster. They will respond faster. Their response time is faster, and so on.
So they know about the 50 cent beforehand, or if there's a feedback that they find out that they did right and they get. I mean, when did they find out how much money they're getting?
This one is actually different. They find it.
Put it at the end.
They've put it in the end. So that's the whole thing with fMRI studies. They change, and it's hard to make the neuroscientists stick to something, because they go by what they actually want to. They go by what they want to test. Anyway, now.
This makes them so it's just going to have a different effect. Right? The fact that it's in the end means that if they start making a lot of mistakes, they see they're not making any money, they're going to concentrate.
Yeah, but
Who
it
knows?
requires a different. It's basically shifting your attention. If you're told 50 cents,
Yeah.
if you find out, and then you get the question does this change?
Hmm?
Does the amount of money change?
Yeah.
So you basically could be expecting, it doesn't matter, I'll get 0 cents, I'll push, because I like crack.
Yeah.
So how is the word determined? What does the word depend on?
Again, it is a randomized. They have pre-selected.
So it's random.
Well, semi-random. They have pre-selected a sequence of words.
But you don't get it. No, but you don't get it if you don't do the right thing.
Yeah. So you get 0 if you make a mistake. Otherwise, you can get 125.
No, they tell you. Not completely randomly. It's predetermined, and for all the subjects is the same sequence.
But from a subject's point of view,
It's random. Yes, you don't know.
So the subject only knows that if
That's
he
right,
or
he
she
could on
does
average
right,
make 25
he gets
cents.
some money.
Yeah,
Right?
could be
It's
0.
It could be 0.
Could be 0. Yes.
So then there's no reason for them to look at the money at all.
No,
Hmm?
there is.
There is.
On average, they're going to make about 25 cents.
No, but it could be 0. But
On average, if they do all trials right.
But it does not depend on whether they do it right or wrong.
No, it does. They don't get money. If they
You can. There will still be activation of the reward system.
They will get 0 for sure if they're wrong.
Yes.
Is that
Yes.
true?
Yes.
Okay.
So therefore, you can make $55 an hour if you do everything correctly. They just don't know how much you're going to make before.
So
they make
why add this complexity? Why not just give them 2 cents or 25 cents every time? they're
Because what you want to see, you mean which complexity?
right? The random reward. Why random reward? Why not?
Because you want to see how sensitive they are to changes in reward.
Oh, I see. And they don't know what the change is going
Yeah.
to be, but so you drive it down and you see whether they stop caring.
Yeah.
I see.
So that's the whole hypothesis, that there is these people, they can still tell reward, okay, but it's not the normal assessment of reward. So gradients of reward are important in
Yeah,
this
but they
case.
don't use that information ahead of time. They just know, oh crap, I made a mistake again. I mean, it seems to not drive the decision.
So anyway, so just from the first task, this is the results I mentioned earlier, where we actually combine the 3 contrast maps just for comparison. Here, drug words, neutral words, drug versus neutral. Okay. This is compared to baseline. This is compared to
What's drug versus neutral? What does that mean?
the contrast map is created to check activations between drug and neutral words. So
How the contrast map computed again? Just the difference?
you pick the scans that you care about, and you see the differences in activation.
Just different.
Yeah.
It's actual minus.
Yeah, because you either compare to baseline or to something else. So here is the relative activations, and you can see that in general.
So what does it mean now that you have DN and 1 to 25?
So here is the behavior for drug-related words only.
Yeah.
Here is the behavior for neutral words only.
Right.
And here is the behavior. This is again the same classification task.
Yeah.
controls versus subjects. Here is the behavior when you're comparing activations between drug words and neutral words. So
you're also comparing 1 and 25.
yeah, and you are also comparing.
How does that work?
Well, so it turns out that drug versus neutral rules. Okay, that's the most discriminative thing, which is something that we expected. What can be compared with the previous experiment is this line here. Okay, and again, the results are fairly similar in the sense that the 0.25 or the 25.50. So that's where you have kind of this relative reward conditions are the ones.
So what does it mean, 125 and that means that
you're looking at
there is 1 cent reward and then case where there's 25
20
cents.
words.
You're also looking at what is drug-related words and neutral words.
Exactly.
And
Together,
then
you're
what
summing them up
do
together.
you do then?
So you
With this,
Hmm?
what do you do with these features? How do you combine them?
you create a contrast map by taking 1 and 25 when there's both drug and neutral. Okay?
That's 4 things. How do you contrast map from what I understood?
So
the difference between two images.
no, you can classify its trial, right? Its scan, whether by a number of things, and then you, depending on how you classify them, you group them. So here's drug and neutral. Sorry, if I said
Is that drug and
Neutral.
union of the words, or is that a particular type of contrast map?
Hmm?
Is D_n, does that refer to a particular way of making a contrast
This
map?
is a particular yes.
Okay. And you have two ways. You have two ways of making contrast
Mhm.
maps. Give us the algorithm from data set that you have for 1 and 25 cents, and all this drug and neutral work. How are you going to pull up data points that you're going to actually?
Don't quote me on this, but I believe that what was done is that you can take, for example, contrasts for drug and 1, drug and 25, neutral and 1, neutral and 25. You can get these groups,
Yes.
and then you can find differences between them. So
there is.
so I'm not sure if first you find the difference between drug and neutral and then 125 or the other way around.
I see. So within those sets, so
Yeah.
you do it twice. So
Yeah.
with this, all four.
So you get one
What we're
data
saying.
point out of all
Yes,
of them.
yes. It's one contrast map,
Okay.
which captures these differences.
Partial derivative works very.
But what's interesting here is that, again, and that was expected, that the drug Q is really discriminative. Okay, we knew that. But again, even in this case, it seems that monetary reward, levels of monetary reward still modify
You said
classification.
The drug queue is most informative.
Well, for all of those conditions, you can see that classification rates go through really jump up once you have drug versus neutral here.
So why couldn't you just use the union of D and then D underscore N and just concat other features together?
You can. And probably if you, in the end, if you want to just do a classifier, that's why we're saying, in the end, if you want to do something that you can sell to companies, for example, to screen employees, probably that's a better test. Okay, but here we're looking at the monetary rewards. This is the thing that's kind of interesting in terms of neuroscience. Okay, and even so the conclusion here is that you can still see significant differences in classification rates between different reward conditions, even when there is the drug versus neutral.
Even when they come after the task.
Yeah. So, and an interesting thing here is that now that we have 5 different conditions in terms of monetary reward, if you actually do a simple majority vote in the end, you will actually get 100% classification, which means that there is no subject which is misclassified in more than 2 of those conditions. Okay, again, that's a kind of encouraging result. But enough with contrast maps. So let me just, because I think I might be running out of time. Problem. I have until 5. It's wow.
You don't want to abuse the patience of everybody, but still.
Okay. So
I mean, we have the room until
Oh, we have the room until 5. Okay, I will try to speed up. So, yeah, there is, the interesting thing is that we can actually use neuropsychological theory and validate it through what I would call an exploratory sort of way. Which leads to the next thing, which is how do we find the most discriminative features, okay? And is time important in finding discriminative features? That's the next question we asked. So now, for each voxel, you get a time series. We're not that good in time series analysis, so we tried a couple of naive things. First, as I've said, we thought of the 3D brain video. The question is, how do we find neighborhoods here and features and so on? So we used the Hilbert curves to come up with a way to linearize the 3D space so that we can get some neighborhood effect. So each 3D scan becomes a vector. Then there is, some dimensionality reduction and feature selection happening, and then classification. Okay. So now, how do we select the most discriminative features? We tried a number of different distance functions, and here is the results that we start getting. So it seems that in the locally reduced feature space, picking up appropriate features, picking up the most discriminative features versus all of the features is actually going to do a little bit better.
And we're back to the first set of experiments. This is
Yeah, this is from now on. The second set of experiments, we're just starting to look. We're just processing it now. So the second set hasn't been published, but that's why we're moving towards a neuroscience submission, because it seems that in the second set, especially for the neutral words, which is comparable to the first one, results are similar. The claims are similar, that you can do classification based on relative activations. Now, if you actually use this time series, they seem to generalize pretty well, and that was pretty much the result that we got out of this. There's nothing really interesting in terms of what we did in terms of the distance and so on. But now that we had a few discriminative voxels, we actually clustered them and showed them to the neuroscientists, and they told us that these regions are actually, one of them is they're actually known to be to have to do with sustained attention and working memory, which is what everybody's doing when they're trying to do the task, and also in processing of serine stimuli and inhibitory control. This is something that my neuroscience collaborators tell me. I don't know. I believe them. And again,
This came out of the time series or of the boosting?
This came out of the time series. This is the stuff that we showed to the.
Why can you only get it out of the time series? Because there's less.
It was just simpler at the time to just find it, cluster it, show it to the neuroscientists. Again, hopefully in a month or so, I will have the final results from boosting from 2 experiments, and I will expect the neuroscientists to tell me if there's something significant there. So now, the third thing that I want to talk about until we kind of all drop is the connectivity and interactivity between brain regions. And I can tell you a priori, this is not the best way to do classification. Okay, the stuff I talked about before does better. But this is interesting. Okay, why this is interesting? Because now you can start actually exploring things that the neuroscientists don't know. And you can actually start looking at patterns that you can show them, and they can start thinking about them. Okay, this is a very preliminary study, and I'm going to use it more because that's going to lead me to a bunch of really interesting questions that I will leave open. So, probabilistic graphical models, I don't think I should explain them too much in this room. Is there anyone here that wants to hear me explain probabilistic graphical models? So, okay.
Yeah, it's getting better. Derivatives.
Well, I started with PCA, okay? Went to boosting, ended up with DBNs. So here, the interesting thing is that there is both parameters and structure, okay? Structure in our case is interactivity between different brain regions. We don't know that.
So in your world, how would you, what's interactivity, what's connectivity, you're going to assume some definition of when the two regions are connected. And
it is functional. It is functional.
It's not ahead of time? Or
no, it's not. So we will start, I'm actually going to,
once we get there.
yeah, I'm okay, I don't need to say why dynamic Bayesian networks here and what they are. So I am going a little faster. HMMs, dynamically multi-linked Bayesian network, BN, not DBN. Anyway, so what you have here is you start with a fully connected network, and you want to learn only the transitions. So here transitions are from one time frame to the next, okay? And these are different brain regions. You have state and observation, okay?
And having a link to imply some kind
Means that there is statistical...
500,000 voxels.
Hmm?
500,000 voxels.
5 regions.
5 regions.
We wish we could do 500,000 voxels, but no, this is 5 regions. Pre-selected by neuroscientists. They told us, we think that these regions, there should be something important going between them. There should be something happening between them.
And these are actually sets of voxels that you're adding?
Yeah, these are neighborhoods. Now we're looking at neighborhoods again, just to make sure that we don't have...
And you want to take a mean of
yeah,
your
8 voxel neighborhoods. So we tried two techniques for structured learning, parameter learning, EM. First hill climbing, then structural EM. So everything's happening in the same EM process. The 5 brain regions, the neuroscientists ran ANOVA. They gave us these 5 brain regions.
And basically, you're saying it's only 8 voxels, so it's pretty well local. It's very local, right? Even though it's 8 voxels.
Yeah. Well,
Small area.
it's these are fairly small areas.
And you can't do this exactly?
Hmm?
Or you can do this exactly?
What?
This learning the structure. You have 5 to 5 transitions, or do you have transitions? Between time steps, you have basically 25 possible.
Possible, yeah.
So you're integrating over everything, or you're not integrating? I mean, is it possible to do this exactly?
What does exactly mean?
You can, but
Yeah.
structural learning.
Yeah, that's funny.
What do you mean do this exactly?
In computer posterior distribution of the hidden variables, you do it exactly.
Well,
Given the parameters, given the structure, you compute the posterior distribution exactly.
Yeah, you must. Okay,
Yeah.
never mind. Let's see what we do.
so let's see. So here is here's the 25 links.
Okay.
Okay. You want the diagonal always to be on, obviously. So the 2 methods actually learn different structures. We don't know which ones. The problem is we don't have ground truth. We don't know which structure, and we don't know if any of them is relevant. The only way to evaluate the structures is to do what we know how to do in my lab, which is classification. Okay. So again, this is, as I'm saying, this is very preliminary work. The only evaluation that we have of those structures is whether we can use them to predict group membership. Again, this is the different links. A little bit more interesting now. is that these are links for controls. These are links for cocaine addicts. And in general, it seems that there is more links between those areas for controlled subjects versus cocaine subjects. What this means is that there is different function, basically.
What are the three? There's the three different subjects.
These are three different subjects, three different control subjects, three
share.
different. And these are the areas that these guys share but are not here, and the red ones are the areas that all these guys share but are not down here. Okay, and the black is the ones that are common to everybody. But in general, what happens is that, and white is links that have been severed. Okay, so what you will see is that there's many more whites here in the cocaine addicts. The way to interpret this is for the neuroscientists is that in these areas it seems that there is not that much interaction. There is a functional change, and for them interaction means probably better function. Okay. So either, as one of the neuroscientists puts it to me, cocaine or drugs short-circuit your reward system. Okay? And so these are some of the effects that we can see of this. Again, do we know if this is happening or not? No. And again, classification, it turns out that we tried all the different parallel HMMs, coupled HMMs, fully connected HMMs, and the two different methods for hill climbing and structural learning for structural EM for our for the networks that we learn. We see that the structural
learning is uniform across time?
It's dynamic.
The structure is dynamic as well. Structure changes.
Position matrix changes across the.
Yeah.
Well, yeah.
Well,
that's way. That is way.
The transition matrices, do they change across, like the links can change from time to time?
In
Or
this-
the same every time that the links.
In
Showing just one set of links.
Yes.
Right?
Yes. No. In this case, we just see them links that seem to be consistent, basically,
Across time.
But at least for the in order to be able to group things and perform classification. So.
Then you don't have alignment problems in that case, right? Because it's uniform.
Yeah, but again, this is getting shaky at this point. So, but again, it seems that our structure EM learning is doing slightly better than hill climbing. So this is for classification, and as you can see, we're only reaching about 85% here. So it's not nice. This is too heavy for classification in terms of computation. You don't need this, but it's interesting because of what other things you can start seeing. And this is related to dynamic causal models, which assume a fixed structure and they just learn parameters. And
how about if you just look at PCA components you were finding before? Do they indicate these links as well?
we haven't tried that. So now here's a bunch of open questions. I'll just throw them here so that they can be food for thought. These are all things we just don't know about. So in our case, we actually used continuous variables. But other researchers that are using this for different brain conditions are discretizing the problem, which makes it much faster. But in general, the trade-off between discrete versus continuous is not clear to us at this point. We haven't done enough experimentation. There are papers that show that structural learning is NP-hard in discrete problems. We haven't found anything. I don't know if there's anyone in this room that knows whether it is whether there are any properties for continuous. There is the question, well, is the fact that you end up using heuristics for when you're doing discrete states, and you end up using heuristics, and you have an error from discretization, is that worth the speed up? We don't know. Static Bayesian nets, where you just find some kind of a network where there is no changes, versus a dynamic Bayesian net.
Yeah.
We actually that's something that we've started looking at for if you don't want to start with pre-selected regions, but you can't run the full dynamic Bayesian network, and you want a guess of which regions to run it through. One thing that we've been looking at, again, this is in the works, is to do some kind of static network analysis, where you train a network on one condition, and then you test it on the other condition. And you want to see which are the regions where there is difference. That means that these are very discriminative regions. These are the regions that you might want to throw in a DBN. That's one first.
Gritty learning on the structure. Essentially gritty learning on the structure where you start adding features. Okay.
Yeah. The question, at some point, we might want to move to event-related designs. And in that case, it's not even clear whether DBNs are going to be the best way to model these things, because now events are not gated in time so well, so the concept of time frame isn't as clear. The whole noise becomes issue becomes much harder. So we don't know. But with these questions, I'm going to try and wrap up. I talked to you about 3 different ways to look at fMRI data: spatial, temporal, and function analysis. This integration seems to be finding some unique patterns of variability, and the hope is that out of all this, one could end up with some objective clinical tool. Up to now, cocaine addiction, once it cannot be detected in standard urine tests after a few days, there is nothing else clinical that can be used. Okay, they just rely on people who come and say, "I'm an addict," or "I'm not an addict." But there is a lot of.
Is anybody who's using it regularly is not an addict? He's not an addict.
No, but whether they're using regularly or how much they're using and so on, this is just by self-report. You can't.
Medical way of determining whether you're an addict.
Yeah. So there is.
Cocaine, right?
So this is for cocaine. So this is something which actually shows physiological difference. That's a good question. I don't know, but it's actually an interesting, a very interesting question.
In experiment with mice, I've talked to people about this before. They showed they have varying levels.
There are varying
rates of addiction.
Yeah, and they are looking even in the subjects that they. No, and the subjects that they are looking, and they have the so-called, which they determine through the psychological interviews and so on, the high functioning or not, they and an interesting thing would be to see, which we haven't done, when they collect more data. An interesting thing to see is whether the high functioning people, in terms of psychological tests, the ones whose behavior doesn't seem to be as affected by addiction, whether these brain changes are as obvious there or not, or whether they form a different category, or whether there is other patterns which compensate. So there is a hypothesis that could be that your reward system, the standard reward system, might be impaired, but the brain develops other ways to compensate for this. Again, this is interesting in terms of neuroscience. So there are a few things that we want to be looking at and be comparing. The one I'm going to mention a little bit more about now is the trade-off between spatial and temporal resolution for fMRI and ERP. ERP is a form of electroencephalography. That means electrical measurements on the skull. Right now, they make subjects where those skull caps, which have a number of probes on them, and I think now the standard is 100 to 150 probes. Now these are instantaneous. Okay, you get something happens in the brain, and you see the potentials change within milliseconds. Okay, and there are certain signals that they know, for example, in the back of the brain, the P300 or something, which they can study directly and draw conclusions about things that are happening. But now, because of this fairly dense sampling on the skull, actually there are reconstruction techniques where they will actually figure out what's happening inside the brain as well. But in general, the spatial resolution of ERP is much poorer. At this point, the people I work with collect separately. Same task. First, they will collect ERP, and then they will collect fMRI. Ideally, they still don't have the appropriate equipment. Ideally, you want to be doing this at the same time. So someone should be wearing the ERP while they're in the scanner, the ERP cap. And then one can start talking about how can one interpolate. And if you think of the spatiotemporal volume of measurements, how can one interpolate? For example, spatial measurements for time slices for which you do not have fMRI.
So how far along? I mean, when is this going to happen? Whether you get something in your
once I get a graduate student that can work on this.
So that actually can be done right now.
Yeah, but it's not going to be that, and that's why I'm not. I mean, it's pretty interesting, and we really want to do this because we think that there is potential. I'm hoping they'll actually get an ERP cap that they will actually start collecting data inside the scanner. Right now, for the drug stroop test, they have the two data sets, but I'm a little worried about registration. It just makes the registration worse, basically. So, and obviously, more studies. Okay, more studies, more experimental data to try these things. I mean, this is just.
But the study is not a problem.
What do you mean?
I mean, it's not a problem for your collaborators to do more experiments.
They are expensive. Their experiments are expensive and time consuming. For them to collect the data.
How much does that cost? The experiment you're talking about, how much will it cost?
Well, apart from the $55 per hour for the subjects, each.
Well, that's if they're really good.
It basically you need you need a lot of people to be working on this. So you need a neuroscientist, either postdoc or a researcher that spends 2 hours per subject, every time that they're going to collect, approximately, if things go well.
How much is time on the MRI machine?
For that particular lab, they have their own MRI machine, so I don't even know how it becomes part of the overhead, so I don't know what that costs. But I think the most important thing is that your standard one researcher, one graduate student type of research team, you might be getting a few subjects per week, basically. Because that's as much as the researcher has time to, maybe they'll do 3, 4, 5 subjects a week. So, and then for all this to be processed, analyzed, and so on, it takes, make sure that the data that they get is valid, because sometimes the data is, the subject was moving too much, and all these things. So it's slow. It took them about a year to collect the second data set that I discussed compared to the first one. But again, I guess it's a question of how much people who do the 200 people studies probably have much more effective and efficient teams. So I will stop here. I think I'm ready for any more questions.
We've asked a ton of questions.
Yeah.
Yeah.
So you mentioned fast Fourier transform to reduce dimensionality. So you're going from the space domain to something else?
No, just. But I mean, you just you go to something else, but you just get the first few coefficients, and that's it. It doesn't really matter that much. You can go back afterwards if you want.
So how many data points do you have in each trial? Like if you have 30 subjects, so you have 30 data points?
Well, what do you mean? You
like
have
in a specific task that you're trying to, a specific condition, like 25 versus 1.
You have, for example, in the contrast map situation, you have 30 different contrast maps.
So you have 30 data points. That's your data.
Yeah. Whatever dimension you pick them to be. Again.
If you kept the same structure, because I'm still sort of thinking about your DBN. If it wasn't DBN, so you showed that there was a full HMM and there was something called CHMM.
Which is coupled HMM.
Coupled HMM. Oh, meaning you separated the regions, so you had an HMM per region. Well, that was the parallel HMM, presumably.
It's just.
Well, let me get together a question. Maybe we don't need to go into detail. How critical is it for you to have varying structure across the steps? Why do you need to have? Do you really need to have dynamical changing structure? Yeah, when you're fitting your series, or can you actually just go ahead and go over all 25? Well, not 25. Sorry. 20, oh no, that's a lot still. 225 possible connections. Yeah, that's a lot still. Okay, you can't do that. Maybe you can.
What?
Can you integrate over all possible? Yeah, never mind. That's too much. Never mind. I don't know. I got my head around it.
Okay. Okay.
Okay.
Thanks. Thanks, guys.